Let me first start off by thanking the Barth Syndrome Foundation and the organizers of the meeting for giving us the opportunity to talk about the work today. And also, my student had an opportunity to give a poster. So for those of you who stopped by and asked her tough questions, thank you, because it means I don't have to do it. Anyway, I also want to thank the families who are showing up. There's some family members sitting in the back, and so thank you again for your support. And I also thank you all for persisting, because it's very humbling for me to give the last talk, because I feel a lot of pressure to maintain the high scientific standards that all of you have developed, and I hope I don't mess it up. So anyway, I'm going to talk to you today about our efforts to develop an enzyme replacement therapy for Barth syndrome and test its efficacy in a mouse model. Very simple in concept, but somewhat difficult in practice. So at this point in the meeting, I don't have to say a lot about background of Barth syndrome. I've removed a number of my slides, but I do have to say a little bit just to show that I know what Barth syndrome is, and that it's a rare X-linked disorder mapping to the Tafazzin gene at XQ28. It catalyzes the remodeling of the mitochondrial membrane phospholipid cardiolipin and helps maintain the CL to MLCL ratio. And I didn't say anything about modification. The CL to MLCL ratio is essential for the optimal functioning of the mitochondrial respiratory electron transport chain. Notice I didn't say remodeled cardiolipin to our colleagues from yeast. And the clinical features are very clear. They include ventricular non-compaction, cardiomyopathy, skeletal myopathy, and cyclic neutropenia. My interest in this field comes from a desire, I am a practicing cardiologist as well as a scientist, is in developing a therapy for Barth syndrome. And again, this is my first Barth Syndrome Foundation meeting, and I'm relatively new to the field. But when I contemplated entry into the field, I was, first of all, it's a very interesting problem, but also a heartbreaking problem. But as I surveyed the literature and looked at what's available, it was striking to me that really no highly efficacious treatment is available. Dr. Byrne, as he mentioned yesterday, is developing gene therapy, which could have a significant impact. But from my survey of what was available, there's certainly no enzyme replacement therapy currently available. And so to me, this represented an unmet need, because as many of you can realize, enzyme replacement therapy has the potential for high clinical impact in affected patients. And there is a mouse preclinical model that we're all using that is available to test potential therapies. As I say, before I leaped into the field, I read a little bit to see, well, what would the obstacles be to Tafazzin enzyme replacement therapy? Well, we would need to be able to make the enzyme we would need to be able to synthesize it fairly easily and purify it. Once we purified it, it must be able to reach and enter affected cells. And so that's actually a big technical problem. How would a soluble enzyme that's inside cells be administered and get inside the cells? And then, as with all recombinant proteins, it must be active after entering the cells. And also, the immune reaction must be limited. And I think those of you in the field will say, we don't worry about this one, because up until this meeting, it's been very hard to make any useful antibodies for many people in the audience. But I think Dr. Claypool's group just developed 3 monoclonals. And so this one I haven't worried about too much. So enzyme replacement therapy is actually here now for other disorders. And there are several on the market for a handful of diseases that are already FDA approved. And if you had a patient, you could go out and buy it. For example, Gaucher's disease and glucocerebrosidase, Fabry's disease and agalsidase beta. There's also an alpha version. Mucopolysaccharidosis 1, 2, and 4 and their respective enzymes. And as we heard from Dr. Byrne yesterday, Pompey's disease and L-glucosidase alpha. And I didn't pay him, but he actually presented this wonderful slide showing a severely affected heart where the patient got enzyme replacement therapy and the heart got much better. All of these diseases are what we call storage disorders, where there's a metabolic defect and these defective substances accumulate throughout the tissues. So these enzyme replacement therapies don't really have to be targeted, so the technical hurdles for these are not as high as there are for Barth syndrome. And that's something we took into account when we started this work. When we started this work, we had the hypothesis that if we could make recombinant Tafazzin, but if we could engineer a cell-penetrating peptide onto it, it would then be able to enter the cells and correct the metabolic and functional defects in these Tafazzin-deficient cells. So a word about cell-penetrating peptides. What do I mean? There's a small group of known peptides that when attached to specific molecules, they can promote the uptake into cells across membranes. They fall into three categories: polycationic, amphipathic, or hydrophobic. And the mechanism of uptake is really not clear, although the preponderance of evidence suggests that most of it is endocytosis mediated, although there are other models and data suggest a direct penetration model or a translocation model through a transient intermediate. There's several well-known naturally occurring examples of proteins that have these cellular penetrating peptides. One is the HIV TAT protein. Another protein is the Drosophila antennapedii protein. I always loved that word, even though I was telling Mindong I've never really been a fly geneticist since I didn't etherize my flies in college and they all flew away, so my Mendelian cross numbers were off. Anyway, and I love the name because in this mutation, the feet actually come out of where the antenna should because it's a patterning defect. The synthetic polyarginines have also been shown to do this. There is a sequence in FGF4 that can also promote uptake of protein into cells. And then another interesting one is called the cardiac targeting peptide, which was identified in a phage display screen for peptides that home to the heart. It's actually not that cardiac specific, but that's the name. Anyway, the two that we've worked with primarily are the Drosophila antennapedii peptide sequence and the cardiac targeting peptide sequence. We tried HIV TAT protein, that particular peptide, but we could never get soluble protein. So our approach is fairly simple. Our goal has been to design recombinant Tafazzin proteins that contain these cell-penetrating peptides, synthesize and purify these recombinant Tafazzins. Then once we've done that, demonstrate uptake into cells. If we can show that they go into cells, then we would need to demonstrate enzymatic activity inside the cells through many of the assays that you have seen here, such as restoration of the cardiolipin modification in the deficient cells, restoration of mitochondrial respiration in the deficient cells. Ultimately, we need to show functional rescue of contraction defects in either skeletal or cardiac muscle. And then finally, we would need to show that we improve the performance in vivo in the mouse model of Barth syndrome. Consider this a work in progress because we don't have a complete story, but what I've highlighted in yellow is what we've been successful at doing so far. And I hope that after we go through the data, you'll be optimistic with me that I think the rest of this is just a matter of time. So let me first talk about our efforts to develop a recombinant cell-permeable Tafazzin enzyme. As I say, the technology is all there and off the shelf. We clone both the mouse full-length Tafazzin and the human version of Tafazzin that lacks exon 5 into a prokaryotic expression vector, which had a polyhistidine tag at the N-terminus to allow for protein purification, and then either an antennapedii peptide sequence, these 16 amino acids at the C-terminus, or the CTP peptide also at the C-terminus. And it was very easy to induce expression, so this is uninduced and this is induced with IPTG, and this band appears, which is our recombinant protein. It's very easy to induce in E. coli, but as many of you who've tried to do this know that it's actually very difficult to make soluble protein. And so, you know, there's sort of a long story to this, and my graduate student has the worry lines, or is developing the worry lines, that it's actually very difficult to make it and get it to be soluble. And we tried all sorts of methods of making native protein and just throwing out the insoluble protein and saving the native, but there's hardly any to use. We tried many different methods of solubilizing and renaturing, and it kept precipitating. But we finally stumbled in, I should say, we scientifically worked out a method where we could actually solubilize the protein and purify it in milligram quantities, which you can see here that just a single pass on a metal affinity column, in this case a cobalt column, gives us highly purified Tafazzin. You know, it's an inclusion body protocol, so we only partially denature. We can renature by dilution into a refolding buffer with containing arginine, and then we can concentrate it on a membrane, and we can get large amounts of usable protein. So it took a lot longer, one slide, but it took a long time to do this. And we've done it for several proteins, several versions of the protein. And so one of the early experiments we did to ask this question of does it enter the cells is we did something simple. We took cultured C2C12 myoblasts and we just sprinkled some protein on at various concentrations. We waited 2 days and we did immunostaining. And as you can see, we used an anti-histidine tag antibody in this particular experiment. And so if we don't add any protein, we see there are plenty of cells there as evidenced by this DAPI nuclear staining, but there's no evidence of uptake of protein, but that's because we didn't add any protein. But then when we added a concentration of 10 micrograms per ml, we could see that the protein was taken up into many different cells. And similarly, with 20 micrograms per ml of Tafazzin, we could see that this protein was going into cells. So this got us very excited. And this also got us funding from the Barth Syndrome Foundation. So again, I thank you. But we went on to show that this recombinant Tafazzin actually localized mitochondria. As our studies evolved, we found that the sigma version of rabbit anti-human antibody, while it doesn't really detect endogenous Tafazzin very well, it can detect our recombinant Tafazzin quite well. And so we did co-localization studies using our recombinant protein without a tag and with a tag, mitotracker dye in red, and then a nuclear stain. And as you can see, when we use a version of the protein which does not have the cell-penetrating peptide, which would be in green, we see no evidence of green fluorescence in any of these cells, whereas the mitotracker shows us the mitochondrial signal in all these cells, and the DAPI shows us the nuclear signal. When we use a version of the protein that has the cardiac targeting peptide, we can see that we do see uptake into the cells. And from the yellow, which is the merger of the red fluorescence and the green fluorescence, we see that there is localization to the mitochondria. So we're very excited so that we can do something that is simple in concept but difficult in practice. We can make the protein, we can sprinkle it on cells, it goes in and goes to the mitochondria. And how about that? Because you know, I like to tell my graduate student, it's rare in your life that things go according to plan. But this is one time where it actually worked. And I said, don't get used to it. Anyway, we've done some corollary methods too, where we've done some biochemical fractionation or subcellular fractionation. And so we've actually taken the mitochondrial fraction from these cells too, and we run them on Western blottings where we've used antibodies to Tafazzin, and we can see the recombinant Tafazzin, but not the endogenous. But again, we also see complex 4, which shows that we actually do have mitochondrial proteins there. And so this is a different cell line, and this is actually the mouse version of the protein, and we've done the immunofluorescence for this too. But I just want to tell you we've done 2 different proteins, 2 different tags, and 2 different methods, and we do see localization to mitochondria. So the important question is, can we assess the enzymatic activity inside cells? And so there are 2 ways that, to do this. One is by doing assessing the tetralinoleol cardiolipin to monolysocardiolipin ratio by either LCMS or MALDMS, and you've seen many examples of this throughout this meeting. And the other is by augmentation of mitochondrial respiration as assessed by measurement of oxygen consumption, and you've all seen many examples by this time. I have to say, our first efforts were focusing on doing LCMS. And as I say, the nice thing about having a graduate student is they do what they tell you until they know better. And so poor Anna has been working very hard to develop the LCMS technique, and we haven't had a lot of success, probably for technical reasons. You know, our spectra just are basically unreadable. And I talked to Bill Poo about a month ago, and he said, why don't you call up Fred Vaz? Everybody else does. And so I said, well, I also talked to Michael Schlami, and he said, well, I'm doing MALDMS. But he also said that LCMS should work just as well, but we didn't believe him. If he's doing MALDMS, we're going to do MALDMS. So we did, and you know, the long and the short of it is, as of last week, Anna was able to get some beautiful spectra that, at least in the heart from the TAS knockdown mice, we see the classic tetralinoleol cardiolipin and the monolysocardiolipin, so we can say that it's working now. We were actually hoping to show data of efficacy of treatment by now, but unfortunately that'll have to wait till we get back to Seattle. So I'm not going to say too much about this, and we recognize it's something we haven't done yet, but it's on our short list of things to do. So anyway, what we do have a lot of data on is assessment of mitochondrial respiration using the so-called seahorse assay, which I think that you've seen many examples of, and I think Dr. Poo early this morning, for those of you who are awake, took everyone through this. But basically, it's an automated bioanalyzer in which a probe is placed into the media of a multi-well plate, and this probe actually senses the dissolved oxygen at various time points, and from that you can calculate the oxygen consumption rate. It also enables you to assess glycolysis, but we didn't really do that. And it also enables you to infuse drugs to inhibit various complexes and characterize the profile of mitochondrial respiration. And as Bill showed this morning, there are several phases. This is what a curve looks like from a seahorse bioanalyzer, in that you get basal respiration, then you get the amount of respiration that contributes to ATP generation, then you get maximal respiration, and then when you finally add entomycin A and rotenone, you get the level of non-mitochondrial respiration. And so that's how we can assess mitochondrial function in our cells that we treat with this recombinant Tafazzin. And before I came to this meeting, I thought it was fairly settled from the literature that, Tafazzin-deficient cells have lower basal respiration and lower maximal respiration, at least as published by Dr. He and coworkers, and also by Dr. Dudek earlier this year, or last year. And, but obviously from this meeting we learned it's very substrate dependent. But nevertheless, back when we were trying to rescue mitochondrial respiration, we went on to make our own C2C12 line of skeletal myoblasts using a lentivirus and shRNA to knock down Tafazzin. And again, substrate is really important. In this particular experiment, we used palmitate as a substrate and found that, at least under these conditions, the knockdown C2C12s have lower basal respiration compared to the wild type, and then lower maximally induced respiration. So the question is, what happens when we add our Tafazzin protein? Do we rescue? And so what we have here, if you look at the red line here, this is Tafazzin plus, this is the knockdown cells plus the wild type Tafazzin that we've introduced. And what we see is that basal respiration is clearly rescued to the same level of the wild type cells. And certainly, if this is the basal level of the knockdown cells, up here we can see that we rescue maximal respiration to the same level as the wild type. We also treated the wild type cells with wild type Tafazzin as a control and found that we really didn't change basal or maximal respiration very much. But this got us excited because this is the first example that we know of to show that exogenous Tafazzin protein can rescue the mitochondrial dysfunction, at least as measured by under these conditions. We've gone on to look at neonatal cardiac myocytes from the TAZ knockdown mice to see if this actually also works in primary cells. And you know, this is not a perfect experiment because those of you who are sharp-eyed will realize that this curve down here is the Tafazzin knockdown neonatal myocytes, and this curve here is the wild type neonatal myocytes. And from Dr. Coutua's work earlier this afternoon, he showed a much nicer curve. See, instead of showing respiration, it looks like I'm showing expiration of cells because these cells are basically not doing anything. But what's spectacular about our recombinant Tafazzin, and the reason I'm showing this, is that under the same conditions, the same cells cultured in parallel, when we give them either the version of Tafazzin with the antenna PDI or the CTP tag, I'm comparing them side by side now, we can see that these moribund cells are rescued and that their basal respiration goes up. In the case of the TAZ knockdown, we can rescue the maximal respiration to reasonable levels. When we take the wild type cells and treat them with either the antenna PDI or the CTP, we get an even bigger boost. And so it says that our recombinant Tafazzin, as I say, this is not a perfect experiment. It was done under glucose conditions. I think that probably our cells are somewhat energy starved, but certainly our Tafazzin can bring these cells back from the dead, which is, again, quite spectacular in our view. Now, what about contraction? Because it may not be enough to rescue mitochondrial respiration. I think if we're to have a clinical impact, we need to rescue the muscle weakness and the cardiomyopathy and also the neutropenia, which, you know, again, I'm just going to deal with the muscle today. One of the ways in which we measure cell contraction in vitro is by using a so-called ion optics system, where cells such as a rod-shaped cardiac myocyte are put on a slide under a microscope, and then it's tracked optically during electrical field stimulation. So when you give the muscle cell an electrical stimulus, it'll contract, and you can measure the shortening of the cell in terms of its cell length, or the decrease in the cell length, and that'll give you a measure of what we call fractional shortening, or how much the cell can contract. We can also look at sarcomere length and how that decreases with electrical stimulation. And if we load the cells with the calcium fluorophore, we can measure the calcium transient. And so this is a standard way of measuring contraction. So we tried to do this with our C2C12 cells, being ignorant. But we determined very clearly that they do not contract in response to electrical field stimulation. So we really couldn't do the rescue experiment in C2C12 cells. And then, of course, we found a paper to show that C2C12 cells in their undifferentiated state don't have any sarcomeres. So we would have saved ourselves a lot of time if we found one of these older papers. Now, neonatal myocytes do contract spontaneously and do contract in an electric field. But the problem with them is their shape. So the ion optics is really optimized for rod-shaped cells because it only interrogates fractional shortening in one dimension. And the other thing that we found in these neonatal cardiac myocytes is a lot of intercell variability in how much they contract, and then orientation under the microscope is a problem. So we actually don't feel that this method is reliable because of the cell shape and significant intercell variation. But we still did it and looked at our data, and we didn't see any obvious difference between the wild type and the Tafazzin knockdown cells. And perhaps that's not surprising because you've heard a lot of data today that the phenotype in these mice, the cardiac phenotype, is not that impressive, certainly not early on in life anyway. And so we've actually carried these mice to 2 months of age. And like other people, we really don't see any difference in ejection fraction or fractional shortening when we compare the knockdowns in red versus the controls in blue. We looked at the mitral valve E to A ratio, which is a measure of diastolic dysfunction, hoping that we might be able to see diastolic dysfunction, something we can measure so that we can try to rescue it with this protein. We also calculated the myocardial performance index, which, as some of you may know, is a measure of combined systolic and diastolic function. And these parameters didn't really differ at 2 months old, and that's consistent with data presented by a lot of other people in this room. So really, there's nothing that we can rescue at 2 months of old, and we may have to wait till 7 or 8 months. And we knew that, but we figured we'd give it a try. We also did ion optics analysis on adult cardiac myocytes isolated from these 2-month-old knockdown mice because many times you can see the cellular defect before you see the organ defect, and so we were hoping that we might see a difference in fractional shortening of the adult cardiac myocytes or diastolic relaxation. We did find that the knockdown myocytes have a significant increase in baseline cell length, but the fractional shortening is not significantly different. I mean, in this particular group, it looks a little bit lower, but that's not statistically significant. And then when we looked at the relaxation time, the time to return to baseline, the curves were basically indistinguishable. So even at the myocyte level, there's no difference at the age of 2 months. So that's something we can't rescue at 2 months anyway. So it looks like, at least in terms of the heart, we're going to have to wait at least till they get to be a lot older and start manifesting cardiac dysfunction before we can try to rescue. Which is hard if you're a graduate student, because if your turnaround for each experiment is 8 months, it means that your PhD is going to take about 10 years, I would think. So anyway, a joke for Anna. Anyway, but what about skeletal muscle? So the skeletal muscle phenotype, as other groups have published, particularly Dr. Burns' group, is that, and Zaza's group, is that skeletal muscle, the phenotype is manifest earlier. So we actually have done myography on skeletal muscle, where we isolated soleus muscle and EDL muscle from these 2-month-old mice. And using myography and measuring the tetanic force generated with high-frequency stimulation, in this case 120 hertz, we see that the knockdown mice have a significant decrease in force generated by the soleus muscle. There's a trend toward decreased force in the EDL muscle, but it was not statistically significant. But we've done this numerous times now. At least we can say this is something we can rescue. So we can take our 2-month-old mice and we can treat them, and we can measure soleus force. Again, we don't have an answer, but at least we have a target. So concurrently, we've been working on ways of delivering this protein to the heart and skeletal muscle in live animals, because that's not a trivial thing, because it has to get there. Even if it can cross cell membranes, you have to get it to the tissue where it really needs it. And Bill is standing up, so I better talk faster. So we have been working on methods of local delivery by direct injection into muscles, by ultrasound-guided injection into the heart, and by endovascular delivery through the coronary arteries. We've also been working with systemic delivery into the retroorbital sinus in mice. We haven't done any tail vein injections yet, but we can. And we have a lot of experience with osmotic mini pumps, because this protein is amenable to loading into an osmotic mini pump and implanting it under the skin and giving a 2-week or 4-week infusion. We've also been playing with biocompatible delivery matrices because as you inject a protein into a muscle, particularly one that contracts, if the muscle contracts, it'll squeeze it all out. So we're actually playing with something called pluronic gel, which is pluronic F127, which has been around for a long time. It has this wonderful property where it's liquid in the refrigerator, but when it warms up to body temperature, it solidifies. So we can dissolve our protein inside, and then when we inject it out of our cold syringe, it'll solidify when we inject it into muscle and won't all wash away. So we've actually used a recombinant Cre protein that we purified and also tagged with antenna-PDI or CTP because Cre is a well-studied enzyme and it has to have enzymatic activity to cause recombination. We've used fibroblasts, mouse embryo fibroblasts derived from the MTMG mice. They're red at baseline, but when the Cre goes into the cells, it'll cause excision of the red gene and cause activation of the green gene. So even within 15 minutes of treating these cells, we start to see green cells, meaning the protein is getting inside the cell and working. Certainly we see it within 30 minutes. We published this, so I won't talk that long. But this helps us to assess our in vivo delivery. And so what we've done is we've taken this protein and we've injected directly into the biceps femoris of these MTMG reporter mice. All cells, all tissues are red until they see the Cre, and the Cre can go into the muscle and cause the muscle cells to turn green. Same with the, when we inject it into the anterior tibial space, it'll go across the epimysium and into the EDL muscle. And when we do vehicle control, we see no background. We also do echo-guided microinjection. So there's an ultrasound of a mouse heart squeezing, and this is the needle going through the chest wall right here. And you can see a little space here where we injected some of our, test injection of Cre in pluronic gel. This is a LAC-Z reporter mouse where the cells that are affected turn blue. I hope you can see that. And I'm almost done, Bill. And the last thing we did is we actually have been working on coronary artery delivery. So we've taken a heart and we've perfused it with Tafazzin, and we've used our sigma antibody, which only detects the recombinant protein, and we can see that we get quite a few green nuclei appearing, up to about 5% of these blue nuclei just with one single intracoronary injection. So we're very excited that we can deliver it. Again, I don't really have any efficacy in vivo to tell you, but I can tell you we're on the way, and I think we're going to have an answer soon. So let me just summarize my work. We've made a recombinant Tafazzin engineered for cellular uptake. We've shown that it localizes the mitochondria and rescues defective respiration. Like other people, we've shown that the Tafazzin adult mice are normal at 2 months, at least from a cardiac standpoint, but the soleus muscles are weak, and again, that's consistent with what's in the literature. What we want to do in the future is we want to show that we can restore the cardiolipin profile. We need to better define the pharmacokinetics. And I can tell you in cell culture at least, a 2-hour pulse will last at least a day. So, and we're doing the in vivo studies to show how long it takes when we inject it, how long it stays in the heart or the skeletal muscle. We need to demonstrate rescue of contraction in the soleus muscle. We need to demonstrate functional rescue of the myopathy and cardiomyopathy in the mouse model, and that probably means that we'll give it to them and we'll do exercise tests because that seems to be a good phenotype, and we'll see if we can restore exercise capacity or muscle strength. Ultimately, we want to partner with the GMP facility to generate clinical grade protein. If this all goes well, we would plan large animal studies and eventually clinical trials. There's also been discussion from a paper Dr. Spiragna published in 2009 that other forms of heart failure also have reduction in Tafazzin cardiolipin. So we're actually testing the ability of our protein to affect other models of heart failure. And we're partnering with an X-ray crystallographer to try to determine the crystal structures of recombinant Tafazzin with the idea that perhaps if we know more about the structure and function, we can develop a super Tafazzin, and which might be better therapy. Anyway, most important slide, as my predecessor said, thanking the people. And I'm a graduate student who's been one of the primary movers, along with Wei Ming, who've done most of the data that you see here. Yonggang Liu has done the ion optics and also does our mouse surgery and heart injections and echoes. Bo helped with the echoes. Mark, Catherine, and Teresa are undergraduates who help with the protein. And Chad is a postdoc who had a different project, but he was very supportive. Nick Aronomakis did the myography for us in Murray Moraes' lab. Dale Whittington in the UW mass spec core has been helping Anna with the mass spec. And certainly thanks to the Barth Syndrome Foundation for funding the initial project in vitro, and then for the coming project on the in vivo characterization. And certainly Matt Toth for giving me a lot of encouragement. Thank you very much.
Thanks, Michael. That's an exciting way to end the conference. Maybe I'll ask Well, I have one comment. Is we also tried our iPS-derived myocytes in that ion optic system, and it also made no difference. I think, as you said, it's the cell shape and the individual cell variation is huge.
Yeah.
I had a suggestion also as a efficacy measure. I wonder if you can see if this thing crosses the placenta and if you can rescue embryonic survival.
We have tried with the Cre protein. So we've taken pregnant MT-MG, because that would be really big if it crosses the placenta. So we've taken our recombinant Cre and pregnant MT-MG mice, and we've injected it systemically, and we have not seen it enter the embryos and cause recombination. So no green embryos. But there's still hope. We can do amniotic injection, because that's, ultrasound-guided amniotic injection, because that would still be very useful.
Right.
And again, every protein is different, so it didn't work for Cre, but we don't know if it worked for Tafazzin.
That'd be a pretty clear-cut endpoint if you could improve survival.
Yeah.
Michael's next talk. I have one comment is that one efficacious experiment you did is that a rescue of the respiration, right?
Yes.
With the seahorse
yes.
test. And in that experiment, maybe I missed something. So you add this Tafazzin fusion protein and you really boost the
yes.
respiration. But did you have a control? For example, is
yeah.
another protein and
right.
doesn't do it.
And we did not do it in these studies, but we actually, as I've shown you from the immunofluorescence, we actually have the untagged protein to do this. We also have mutated protein where we've engineered the some Barth syndrome mutations in to see if they don't rescue, but we haven't done it yet. But we agree with you, we need to do a inactive protein control, either missing the cell-penetrating peptide tag or having a Barth syndrome type mutation. So we're going to do that.
Okay.
Hello, I have a question. Actually, I have two questions. The first question is, what's the efficiency of you knockdown in the C2, C12 cell lines?
I'd have to ask Anna, but I think it's about 80%.
80%?
Yeah, I believe so. And she did the QRT-PCR, so you can bother her later, but my memory is it was 80%. So she's nodding, so I think I'm right.
So the mRNA level is 80%?
mRNA level is 80%,
and
yes.
Did you measure the protein levels?
No, we haven't, but we should. But yeah. Again, our antibody doesn't detect endogenous, so it's actually been difficult for us, but maybe if Dr. Claypool will give us some of his monoclonals, we can do that.
Okay, and another question is, did you measure the other phenotypes that best cells should have, like the CL profiles, the supercomplex in the C2, C12 knockdown cell line?
No, we haven't done that because, again, I think we're just trying to test efficacy of therapy because I think that, nobody would ever mistake me for a mitochondrial biologist. My original training is in transcriptional biology, so nobody would ever expect me to do it or think I would be any good at it, but it's a good experiment, but we haven't done it. I think it'd be very important to do that.
Thank you.